Good evening, sir.
Good evening, ma'am. Good evening.
You can start, sir.
Okay, ma'am. Thank you so much, ma'am. Good evening, my dear students. Today, what we are going to discuss about, the Hardy-Weinberg law. Already we saw it in the introduction part, isn't it? What is the main uses of the Hardy-Weinberg law? How it will be constructed? What are the main important of this law? Okay, and how they are calculating? For example, homozygous, heterozygous, and also recessive aspect as well as the dominant aspect. That things how they are calculating. Only these equations. There is one of the equation already is studied in the max aspect. Okay, already you know about a plus b whole squared, isn't it? Okay, similar to that one. Here, the Hardy-Weinberg law also they were used for the identification of the behavior of the genes in the population. Suppose if you want to know the particular genes present in the previous or ancestor population, how will you identified? Okay, so that time they were used the Hardy-Weinberg law. See here, before that one, behavior of the gene in the populations. There is a common misconceptions that characteristics associated with the dominant allele. Normally, about each and every characters is also related to the dominant alleles only. Suppose the dominant alleles is increases in the frequencies because of its genetic dominance. Okay, for example, father has a 50% and mother has a 40%. Which one is the dominant? Father is the dominant, isn't it? 50 alleles also, 50 genes also inherited to the one generation to another generation. This misconception is refused by the principle called as Hardy-Weinberg equilibrium. Already we saw it. This was first introduced by in the year 1908. C.H. Hardy shows that in the population in which random matching occurs and in the absence of the force that changes the proportions of the alleles, the original ratio of the dominant alleles to recessive alleles is retained from one generation to another generation. How will you calculate that? Which one is a dominant allele? Which one is the recessive alleles? Because in previous ages, there is no any proper, what is that? Matching system, isn't it? Randomly they are mated, isn't it? That's the reason. So, which gene is a transfer from the which area, which place? How will you identify? Means the one of the these laws only. So that is the reason. The behavior of the genes of the population we can easily calculate with the help of the equation is called as Hardy-Weinberg law. It can be, what is that? Misconception is refused by the principle is called as Hardy-Weinberg equilibrium. Okay, see that one. So to demonstrate this, they examined the behavior of alleles in an idealized non-evolving population in which 5 conditions hold. Suppose if you want to demonstrate these things, have to, what is that? 5 condition is required. 5 condition is followed by the scientist. One is called as no mutations, isolation from other populations, large population size, random matching, no natural selection. These are the 5 conditions is followed by this Idealization, non-evolving populations. It will be possible or not? We do not know. Okay, how will you identify? These are the equations. By the process is called as what is the Hardy-Weinberg equation? That Hardy-Weinberg equation already you studied. So the Hardy-Weinberg equation, that is equal to P plus Q whole square, that is equal to 1. This is the normal range. Okay. Suppose the 1 is the result, isn't it? Suppose the 1 is the result. Think about this one. Suppose it is less than that of 1 or more than that of 1. What happened? The chromosomal aberration, chromosomal gene, chromosomal mutation also occurs. Okay. So the net result is 1. This is the equation. This is the result. Okay. It can be explained, the expansion aspect already know P squared. So here, P plus Q whole square, it's also expand in this format. P squared plus Q squared plus 2PQ, that is equal to 1. That is equal to 1. Just imagine. What is the meaning of P squared? It's nothing but the dominant alleles. Dominant alleles. Take an example. Capital A, capital A. Okay. This one is called as what is that? Recessive alleles. Take an examples of small a, small a. Okay. Both are homozygous, but at the same time, this one is a dominant. This one is the recessive. Okay. What about this one? This one is a heterozygous because of both alleles present in 2PQ. Okay. This is the way to identify it. Suppose these are all what is that? Equal to 1 only. Suppose there is a variation occurs in this result or net result, it will lead to a mutation. Don't forget, my dear students. Okay. See that. See that in this equation, P squared plus 2PQ plus Q squared, that is equal to 1. Already we saw it. What is the meaning of P? P is designated the frequency of one allele, which is the one allele at a specific locus. Already you know, but what is by locus? What is by locus, my dear students? Locus is nothing but a particular location of the chromosome, isn't it? The allele present in the particular location is called as locus. Okay. Here, for example, dominant alleles, A is a dominant, isn't it? When you are putting the dominant character, capital T is also a dominant. In this category, capital A is a dominant character. What about the Q? See that designated the frequency of the other allele. Other allele means what is the opposite of the capital A, opposite of the dominant. Which one is the opposite of the dominant? The recessive. It is denoted as a small a. Okay. These are the two things. Okay. Two alleles or dominant character, recessive character also occupied in this portion. Okay. So in this the equation. See here. P to designate the frequency of individuals homozygous dominant. Already told you, homozygous means, see that capital A, homozygous, both are homozygous is nature. Same alleles present in the particular locus is called as homozygous. Q2 is the frequency of individual homozygous for the recessive, the other alleles, recessive of the other alleles. This is a small a, small a. This is a recessive character. Okay. What about 2PQ? So these are all comes into the equation. P squared and plus 2PQ plus Q squared, that is equal to 1 is the equation. The Q2 is the frequency of the individual homozygous recessive the other allele, small a, small u. 2PQ is the frequency of heterozygous. Because why it is called as heterozygous? The dominant also allele also present in it, and recessive allele also present in it. In this way, just listen, my dear students, very, very important. In this way, the max calculation must come in your question paper. Don't forget. This is very, very important part. Don't forget, don't forget. Okay. See here. So jump we will see that this equation is also how it will be used for the max aspect. Okay. I will show you in the last one. Suppose if we want this one, just go to the see the last portion, we can easily identify. One of the example has I have taken from the see here, this is the question. See here, this is the question. In a HW population, just listen here, my dear. HW population of 1600 individuals. From this one, 250 are showing recessive triads. What is the meaning of recessive? Recessive denoted as a small a was given. Otherwise called as Q squared only given here. You have to find out which one, heterozygous condition. Which one we have to find out, 2PQ you are going to find out. Okay. From this individual population. How will you find out? It can be mentioned. It can be calculated by a small, what is that? Video format. Just to see it, you can identify the answers. Okay. Even you get that. I think it is not coming. Okay. I will show you. Okay. I will show you when listen here. Are you seeing, Pa? My dear Ajay, are you seeing this way?
No, sir, I can't see, sir.
You couldn't see. See, listen here. I will show you. Okay. Just a minute. Okay, anyhow, just say I will show you after sometimes, okay? Because it's not visible. Here I have taken, but it's not visible. Okay. See here. So this is the equation already saw it, isn't it? Okay. So the P squared plus 2PQ plus Q squared that is equal to 1. P is designated frequency is 1 allele at the specific locus is dominant A and Q is a recessive, isn't it? See here. P squared. So this is the P squared. Okay. A. One is considered as a P, another one is also P. So it is also P squared. Q squared, it's nothing but recessive allele A at A. This is also one Q and another A is also another Q. So that's why it's a Q squared here. 2PQ. What is the meaning of 2PQ? You have to put here 2. 2A and 2Q also is that. Okay. How it will be calculated. You will see the things. You can last, I will show you. Okay. Some genetic disorders. What are the genetic disorders present in it? Genetically modification. Already we studied so many times. Again and again, it repeated for your book with the examples. Okay. Phenylketonuria. It is very, very important. Once the phenylketonuria is a rare metabolic disorder that affect the way the body break down protein molecules. Phenylketonuria already we studied. How it will be? One of the enzyme secretions, which chromosome is very, very important one. The phenylketonuria is caused by the mutation in gene on chromosome 12. Note it down, my dear. Here. So phenylketonuria, which chromosome disorder is? Excuse me. Can you visible, Ba?
Yes, sir.
Okay, just Hindi.
इस प्रिंसिपल के अनुसार किसी पॉपुलेशन में अगर एलील्स की फ्रीक्वेंसी जनरेशन टू जनरेशन चेंज नहीं हो रही है, यानी समय के साथ एलील्स की फ्रीक्वेंसी में ज़्यादा चेंज नहीं आ रहा है, वो स्टेबल हम कह सकते हैं। तो ये पॉपुलेशन हार्डी वाइनबर्ग इक्विलिब्रियम में हालांकि पूरी तरह से इक्विलिब्रियम में तो कोई भी पॉपुलेशन नहीं हो सकती नेचर में, क्योंकि नेचुरल सिलेक्शन से हर समय पर एवोल्यूशन की प्रोसेस चलती रहती है। कुछ पर्टिकुलर एरियाज जो कि बेनिफिशियल होती हैं, फिटनेस एनहांस करती हैं, और समय के साथ सक्सेसिव जनरेशंस में उनकी फ्रीक्वेंसी बढ़ जाती है। लेकिन क्योंकि ये बहुत ही एक्सट्रीमली स्लो प्रोसेस है, ओवर थाउजेंड्स ऑफ ईयर्स, मिलियंस ऑफ ईयर्स एवोल्यूशन होता है या लाइफ फॉर्म्स में कोई विजिबल चेंज आता है, तो इसीलिए हम पॉपुलेशंस को उनमें एलील फ्रीक्वेंसीज का कैलकुलेशन करने के लिए लेके चल सकते हैं कि हाँ अभी ये फिलहाल स्टेबल है, इनमें इक्विलिब्रियम मेंटेन हो रहा है, क्योंकि अचानक से बहुत कोई बड़ा चेंज आ नहीं रहा। तो इस तरह की पॉपुलेशन हमने दी हुई है, हम मान एक पर्टिकुलर जीन के लिए लेके चलते हैं, इसके हमारे पास दो एलील है, पी हमारी सपोज डोमिनेंट एलील है, मेंडल के हिसाब से ही चले तो कोई भी जो कैरेक्टर है दो एलील्स के कंट्रोल के अंदर होगा, एक जीन के स्लाइटली डिफरेंट फॉर्म्स होते हैं, इनमें से एक डोमिनेंट होगा जो हमेशा खुद को एक्सप्रेस करेगा, और रिसेसिव वाला जिसको क्यू लेके हम चल रहे हैं, ये सपोज डोमिनेंट वाले के आगे सप्रेस हो जाए। तो यहाँ पर तीन तरह की कंडीशन हो सकते हैं पॉपुलेशन में, या तो ये हो सकता है दोनों पी यानी डोमिनेंट होमोजाइगस कंडीशन, हो सकता है दोनों क्यू डोमिनेंट रिसेसिव कंडीशन, और ऐसा भी हो सकता है कि हेटेरोजाइगस हो, यानी एक पी और एक क्यू, एक डोमिनेंट और एक रिसेसिव एलील किसी के पास में। तो इन तीनों के अलावा तो कोई पॉसिबिलिटी नहीं है डिप्लॉयड ऑर्गेन्स में, या तो दोनों पी होंगे या तो दोनों क्यू होंगे या पी क्यू होगा, और इसके अलावा कोई और एलील दो ही एलील हम लेके चलेंगे। तो ऐसे में इनकी फ्रीक्वेंसीज को अगर ऐड किया जाए, इस जीनोटाइप की फ्रीक्वेंसी को हम पी स्क्वायर मान के चलते हैं, फिर इसकी फ्रीक्वेंसी को हम क्यू स्क्वायर मान के चलते हैं, और हेटेरोजाइगोट की फ्रीक्वेंसी हम ले लें टू पी क्यू। तो इन सारी फ्रीक्वेंसीज को आपस में ऐड करके हमको बराबर 1 मिलना चाहिए, क्योंकि पूरी पॉपुलेशन इन्हीं तीनों पॉसिबिलिटीज की बनी हुई है, इसके अलावा तीसरी तो इसके अलावा चौथा तो कुछ है नहीं। अच्छा, इस एक्सप्रेशन को जब देखते हैं तो ये हमें a प्लस बी के होल स्क्वायर की तरह नजर आ रहा है, क्योंकि a प्लस बी का होल स्क्वायर भी यही होता है, a स्क्वायर प्लस 2ab प्लस b स्क्वायर। तो इसको अगर हम इस बायनोमियल एक्सप्रेशन को शॉर्ट करके लिखें तो p प्लस q का होल स्क्वायर बराबर 1 लिख सकते हैं, और यहीं पर अगर दोनों साइड पे स्क्वायर रूट ले लेते हैं तो p प्लस q भी हमें बराबर 1 मिलना चाहिए। तो यहाँ में हमें बोल क्या रहे हैं कि हमारा टोटल जो है 1600 है, जिसमें से 256 रिसेसिव ट्रेट शो कर रहे हैं। रिसेसिव ट्रेट शो करने वाले कौन होंगे? रिसेसिव ट्रेट वही शो करेगा जिसके पास होमोजाइगस रिसेसिव कंडीशन हो, क्योंकि हमारे हेटेरोजाइगोट्स और हमारे डोमिनेंट होमोजाइगस इनका फिनोटाइप तो डोमिनेंट वाला लॉ ऑफ डोमिनेंस के अनुसार। रिसेसिव ट्रेट रिसेसिव वाला फिनोटाइप खाली ये शो कर सकते हैं जिनका जीनोटाइप ये हो और फ्रीक्वेंसी उसकी क्या है क्यू स्क्वायर। तो यहाँ पे 256 हमको इनकी फ्रीक्वेंसी दी हुई है, अगर हम परसेंटेज के हिसाब से निकालें तो 256 1600 में से कितना होता है, ये होता है इसका 16%। तो 16% इसकी फ्रीक्वेंसी हमारे क्यू स्क्वायर की इस टर्म की। तो अगर क्यू स्क्वायर हमारा 16% कितना हो गया 0.16 हो गया 16% का मतलब, तो क्यू हमारा कितना निकल के आएगा, स्क्वायर रूट ले लें इसका 0.4, और यहाँ पे p प्लस q बराबर 1 है, तो अगर q हमारा
Sir, you are not audible, sir.
Yes, ma'am. Yes, ma'am. No, ma'am. The video is going on.
बताना क्या है, अगर इतने रिसेसिव ट्रेट शो कर रहे हैं तो हेटेरोजाइगस कितने होते हैं, यानी हमारी हार्डी वाइनबर्ग की इक्विलिब्रियम की इक्वेशन जो हमने
Can you hear my voice? The video voice, ma'am?
Now I can hear you.
Okay, ma'am. Okay.
इसकी फ्रीक्वेंसी के लिए हम ये टर्म यूज कर रहे थे 2pq, तो हमें q मालूम चल गया, p मालूम चल गया, हमें खाली 2pq बताना है। तो ये हमारी हेटेरोजाइगोट्स की फ्रीक्वेंसी क्या आएगी, 2 into p into q। तो परसेंटेज के हिसाब से ये कितना बन गया 0.48 हमारा आंसर आ रहा है, यानी 48%। तो किसका 48% हमको लेना है, टोटल पॉपुलेशन हमारी 1600 बता रही है, हमें तो ये फ्रीक्वेंसी हमारी निकल कर आई 2pq जो निकल कर आया, तो फ्रीक्वेंसी हमारी 48% है। तो अगर हमें कितने इंडिविजुअल्स ये शो कर रहे हैं हेटेरोजाइगस कंडीशन बताना, तो हम 1600 का 48% ले लेंगे, जो कि हमारा कितना निकल कर आ जाएगा, ऑप्शन ए 768। तो हार्डी वेनबर्ग पॉपुलेशन अगर हमसे कह रखा है तो इससे हमारा मतलब होता है कि इसमें एलील
Okay. Can you see the video, Ajay?
Yes,
No,
sir.
Do you understand that one? Do you understand that one? You can't, isn't it?
Sir, video was coming, sir.
Sir, video, no, sir.
Not it, not at all. Okay, I will
Okay.
explain it lastly. No need to worry. Okay. Okay. Listen here.
Yes.
Yeah. Right. Some genetic disorder already we studied. So what is the first one? Phenylketonuria. The phenylketonuria is nothing but the one of the protein breakdown. Already you know very about. So which chromosome is responsible for this one? 12th chromosome. Okay. The absence of the enzyme is called as phenylalanine hydroxylase. Okay, in the liver, these are the important one. Suppose the phenylalanine hydroxylase secretion is not possible. What happen? Automatically the conversion is not possible. Already you know all about. The enzyme break down the amino acid phenylalanine into other product that is called as tyrosines. Already discussed. So it is one of the autosomal disorders. This is also very, very important one. Okay. So you must note, first was phenylketonuria and 12th chromosomes, which enzymes, and what will be the product. Okay. So it is also one of the autosomal recessive disorder. Okay. See that one. Dietary protein. So phenylalanine is also isolated. The phenylalanine is one of the amino acids. It's also converted to tyrosine with the help of the enzyme is called as phenylalanine hydroxylase. Suppose the enzyme is absent, so what happens? The conversion is not possible. So as a result, phenylalanine deposited in the blood. It will not excreted through the urine, so it will be deposited in the brain also. Sometimes it will lead to some other difficulties. Already we saw it, that things also. Okay. Okay. See here. So alkaptonuria. It's a second one is that. Okay, this is also same. It is otherwise called as black urine diseases, my dear student. Don't forget, my dear, black urine disease. The alkaptonuria is an inherited condition. What a cause of urine to tan black when exposed to air. A particular person suffered by the alkaptonuria, his urine or what happened, when it exposed to the sunlight, what happened, the pale yellow color urine is also tan into black color when exposed to air. Okay. So there is a, what is happening, there is a absence of homogeneous state oxidase enzyme, which break down the amino acid phenylalanine and the tyrosine. There is an accumulation of homogenistic acid in the body. Note it down, my dear, the homogenistic acid. So due to the conversion, what happened, the accumulation of one of the acid, homogenistic acids deposited in the body. As a result, what happened, the urine conversion will be takes place where pale yellow into black color when it exposed to the atmosphere. Okay. See that this is the symptom and sign of the alkaptonuria. Black colored urine. So when it the urine is collected, that time it will be pale yellow in color. After that, what happened, it exposed to the atmosphere. What happened? It tan into pale yellow to black in color. And also another one is called as achronosis. So see that the mouth part will be like this. There is a small, what is that? Patches. Okay, small patches just like a wound or pimples like structures formed the mouth parts only. Okay. Another one is also here, the leg. And third one is kidney and also the prostate stones also formation takes place. See that this is the kidney structures. So inside there is a white part is there, no? So this is nothing but what is that? Stone formation takes place inside the kidney due to the deposition of the acids. And also another one is called kyphosis, or another one is called as UNCAC. It's another type of disease also mostly affected by the central nerve systems. These are the symptoms due to the alkaptonuria. Okay, see that one. So this is alkaptonuria. See here, this is the normal urine. It may converted into exposed to, when it exposed to the air or atmosphere, what happened, it may be turned into a black in color. Okay. That condition is called as alkaptonuria. How it will take place? The conversion of phenylalanine into tyrosine with the help of the enzyme is called as phenylalanine hydroxylase. Okay. Due to the lack of enzyme, what happened? Already we discussed. See that. Another one is the very, very important one, my dear students, already we studied. Turner syndrome. The Turner syndrome. See here, this is the 46 chromosome, isn't it? 45 chromosome plus X0. So this is a chromosomal format or chromosomal numbers of Turner syndrome. Don't forget, my dear. This is already so many times they were asked in the questions. Turner syndrome is a chromosomal condition that affects development in females. Okay. Turner syndrome mostly occurs in the development in the females. That means, what is the meaning of, when the Turner syndrome happens, what happened? See one by one. The most common feature of Turner syndromes in short stature. The ovary develop normally at first, but egg cells, oocytes usually die prematurely. That condition lead to the sterile condition of the female. Okay, don't forget, my dears. The ovary develop normally at first. First it developed. After that, what happened, but the egg cells, oocytes usually die prematurely. People with Turner syndrome have an extra fold of skin on the neck. That is called as webbed neck. Don't forget, my dears. What will be the symptom of the physical aspect? Phenotypal aspect means you have to note it down. The particular person, those who are suffered by the Turner syndrome, the skin will be, what is that? Webbed. This extra fold. See that? You can see the diagram. So this is the chromosomal number of the Turner syndrome. Already in our board. How many pairs? 23 pairs, isn't it? 23 pairs. So 22 means what is that? Already we studied it. 22 means 44. Here 45 plus X0. So this is the symptom already we mentioned. So these are the 44 chromosome already present in your body. One is a sexual chromosome, isn't it? The sixth chromosome is absent. One chromosome is absent here, isn't it? So that's why it is also 0. So the female, what happened, lose of one, what is a chromosome? So 45 into X only. That's why it's X0. They mentioned in the title. See that? 45 plus X0. Okay. This is the symptom. So the chromosome number. See here, already but already we discussed. The female, the 46 chromosome is the somatic cell. Male also 46. During the gamete formation, see that this is a homozygous. So they will produce 2 X chromosomes or gametes. So each one it carries 23 and here also here 23. What about the XY? 46. So sometimes it may be divided like this unexpectedly. 22 into 24. So the male gamete X or female gamete of X fused together, you will get it this type. Okay. 22, 23. 45 you will get it. So X0 is also possible. On the other hand, what happened? The another gametes also 23 is there, no 50% of the female. It's also joined with the another one is of the 24 unexpected divisions. You will get it 47. This is 44 plus 3 XXY. How it will be already? We will discuss one by one. This is the separation of the gametes during the gamete formation. Also fusion takes place at the time of fertilization. What happened? How it will be divided? That things also already discussed. See here. So this is the Turner syndrome. See that 45 DXO. See the short stretchers, small mandible, high anchor plate, low set of ears, low hairlines. Okay. The webbed neck. Webbed neck means, see, the neck is just like a duck. And there is a muscles also present in it. That's why they have given as a webbed neck. Already we discussed. Okay. Bicuspid aortic valve coruscations of aorta. So these are all that we do not know how to. The minimum, see that the female mammary gland is also formed. Okay. The diagnosis of karyotype, this is another type. The particular chromosome will be removed. Ovarian dysgenesis, amenorrhea, infertility. Okay. The female character is there, but infertility natures. Okay. This is also very, very important. Turner syndrome, look like this one. So due to the 45 into XO, X0, like that. Okay. See that one. And next disease is called as Lesch-Nyhan syndrome. Lesch-Nyhan syndrome is a condition characterized by a accumulation of uric acid in blood in urine. Some of the persons we were analyzed, we can see some of the persons, they are having the knee pain, okay, due to the accumulation of the uric acid already in our body. Uric is also one of the protein derivatives. Okay, protein is also the protein amino acids also converted into urea, uric acid, creatine, creatinine. Already we studied. They are all stored in the liver, and also they can mix with the blood, and also it can be filtered by kidney. Suppose if it is not possible, what happen? Automatically it will circulate throughout the body again and again. What happen? It will lead to some disease or syndrome. Okay, see here. So the Lesch-Nyhan syndrome is nothing but the uric acid in blood in urine, which can cause gouty arthritis. Arthritis is nothing but the knee pain, kidney stones and bladder stones. These are all due to the formation of the uric acid. Sometimes it may be a stone. Calcium oxalate is also formed. Okay. Self-injury, including biting and head banging, in the most common and distinctive behavioral problem. This condition is inherited in an X-linked recessive pattern. X-linked recessive pattern. Okay. This is very, very important one. Okay. So you must note it down. Lesch-Nyhan syndrome, uric acid. What will be the symptom? So particularly the arthritis, gouty arthritis, kidney stones, bladder stones, all are related to urinary problem. Okay. Which is the behavioral one? So which chromosome is responsible for this one? No, X-linked recessive gene only. Okay. See that one. See, the symptom is look like this one. See the fingers, there is a nail is there. Then see, this condition is when you are seeing the any one of the person, that person suffered by, what is that? Lesch-Nyhan syndrome. See the nail. Okay. So this is the symptom. And mnemonics, Lesch-Nyhan syndrome is also L first letter and second one N and also S. Lack nucleotide salvage. Okay, nucleotide means that is nothing but the purine molecules. Purine. What is by purine? Adenine and guanine is the purine molecule, isn't it? Pyrimidine, thymine and cytosine. These are all purine. Pyrimidine, sorry. So the purine content, so it will happen. Suppose if it is produced by the blood in the form of uric acid. And the next one is called as Klinefelter syndrome. Note it down, my dear. This is also very, very important. The Klinefelter syndrome mostly occurs in the male sexual development. See, that Klinefelter syndrome is a chromosomal condition that affects male sexual development. Male with these conditions typically have small testes and that do not produce enough testosterone. Okay, this is the Klinefelter syndrome. The shortage of testosterone during the puberty can lead to the enlargement of the, what is that, mammary gland. The enlargement of the mammary gland in male, that condition is called as, what is that, gynecomastia. Sorry, gynecomastia. Sorry for the inconvenience. Gynecomastia. Reduced facial and body hair and the inability to father. Note it down. Children. Infertility. The Klinefelter, that is denoted as, what is that? Two things is there. They do not produce enough testosterone. Mammary gland development, that condition is called as gynecomastia. Inability to father, that is infertility. Okay. So these are the symptoms. See that Here what happening? The same thing is that 22 pair as it is. Okay. The last one, what happened, see that the XX, XY is that. The XY is the male character, but the one addition of one X is also here. Okay. So as a result, what happened? The male, he has, what is that? Female X character extraordinarily. As a result, mammary gland formation also takes place, but they will not produce, what is that? Enough testosterone molecules. And finally, the child is not possible. They cannot be able to, what is that? Fertile the egg. So this condition is called a C. Note it down, my dear student. This is very, very important one. XX, XY, X, double X, Y. Okay. So these are the symptoms. And then Down syndrome is the another one. Down syndrome is a development of disorder caused by a extra copy of chromosome 21. Note it down, my dear student. 21. Okay. Down syndrome, the extra copy of the chromosome 21. The people with the Down syndrome have a very distinct facial features. This type of those who are suffered by the Down syndrome, his face or her face, what happened? The different facial features, and flat face is there. A small broad nose is also there. Abnormally shaped as a large tongue and upward slanting eyes with a small fold of skin in the corners. These are all the symptoms of which one, Down syndrome. See the diagrams, see the slide, you can easily identify it. See, these are the peoples. Already we can saw it. Already we saw it. Okay. So Down syndrome, see that small head at birth, abnormal ear. Yeah, see the ear is very large in size. Okay, flattened nose and also face. Faces look like a Japanese. It's not exactly. So look like a Japanese. And white spot on the colored part of the eye. Okay, see, and then slanting eyes is also. See that hands and foot also look like this one. Okay, it is not well developed. See that hands. Okay, so look like, see that eyes also look like, nose also. Okay, so these are the symptom of which one, Down syndrome. Which one is responsible? The chromosome of 21. See here, the extra chromosome activity. 21, there is a copy of another one, X1. 21st chromosome is also, what is the additional one chromosome is added into the 21st one. So as a result, the symptom is possible. This is also one of the polyploidy nature. That's why it comes under the heading chromosomal abnormalities. Okay, so this is the another one. See that 21 chromosomes. Trisomy nature. Note it down, my dear. Trisomy. Trisomy means, what is the meaning? Already you know well about. So okay. So what is it meant by trisomy? See that one. Already we saw it so many times. Okay. See though. What is my trisomy? See, when the chromosome is trisomy, what is the meaning of trisomy? You know well about N. Okay, so N plus 2N, that is equal to 2N, that is equal to 3N. This condition is called as, what is that? Trisomy, isn't it? Okay, so what about the nullisomic means? What is nullisomy means? 2N minus 2. So you will get it at 0, isn't it? This is nullisomic. Okay. So what about the monosomic? Monosomy 2n minus 1, you will get 1 only. So this is the monosomic nature, isn't it? This is the monosomic nature. So these are all very, very important. But in this category, what they have, the 21st chromosome, what we are having 3 types is that 3 chromosome is there. Okay, so 3 chromosome present in the which place? So 21st position. Okay, these are all very, very important, my dear students. Okay, go to the next one. Okay, so this is the Klinefelter syndrome. Already we saw it. Down syndrome also we saw it, isn't it? So, and then Tay-Sachs disease. Another one is also here is that these are all are very, very important, my students. These are all are comes under the headings known as what is that? Genetical disorders. Isn't it? See the Tay-Sachs disease. It's a rare disorder. Okay, this is a rare disorder. Note it down, my dear students. That progressively destroy nerve cells. Note it down. Tay-Sachs disease also here. Destroy the nerve cells and the brain and the spinal cord. There is absence of the enzyme beta hexaminidase A. Note it down, my dear students. This part is very, very important. What is that? So beta hexaminidase A, which play a critical role in the brain and the spinal cord. What is the main function of this? You can see it. So it is very, very important, the brain function as well as the spinal cord. It's related to the nerve system. Tay-Sachs diseases. Okay, what happened here? See here. So this is very, very important. Tay-Sachs disease. Suppose what happened, suppose if the enzyme is not secreted, what will happen? The particular functions of the nerves as well as the brain, it will be affected. So as a result, sometimes maybe strokes and the blood clotting mechanism is also possible in the brain. See here. So Tay-Sachs diseases, the hexagene mutation is a 6th chromosomal thing. 6. Hexa means 6. Swollen neurons with lamellar inclusions due to the accumulation of the gangliosides and demyelinations. Demyelination, removal of the myelinated seed from the, which one cell? Nerve cells. Okay. Demyelinate. So the mental and the motor delays. This one is also very, very important. So the improper, what is that? Impulse passed from the central nervous system to peripheral nervous system. Sometimes the peripheral nervous system to central nervous system. These are the Tay-Sachs diseases. See that one? Another one also. So this is the things, accumulation. Okay. So in the brain cells, okay, what happened? It will lead to the mostly the child may affect mostly. Okay, see that. So this is the calculations. Already we saw it in a whole double populations of 1,600 individual, 256 or so. You have to calculate here, So 256 divided by 1,600, what will happen? What will be the percentage? This is, see here once again. Read the question carefully. In a HW population of 1,600 individual is there. From this one, 256 are showing a recessive triads. 256 are recessive. Recessive means what is that? Just to apply the Weinberg law. P squared plus 2PQ plus Q squared, that is equal to 1. The Q squared is 256, isn't it? Recessive. Recessive is 256. So 256 divided by 1,600 into 100, you will get it. What is that? 16% you will get it. So I will show you. So that means it is not in the. So this is another one. Can you see the video, Ajay?
No, sir, it's not playing, sir.
Not playing now. So how can I? Okay, the calculation I will put it in different manner. Okay. So I think that's all about the class. That's all about the things today. See that. Okay. See, yeah. This is the question, isn't it? Can you see the board, my dear?
Yes, sir.
Okay. So how many percentage is there? Just to draw the 1600 is the individual population, isn't it? Okay. So what will be the recessive triads? 256 only, isn't it? Recessive means what is that? Q. Okay. This one is total. Okay. Total. So what will be the equation? You know, P squared, that is equal, sorry, P squared plus Q squared is equal to 1. Okay, so it is not okay. Anyhow, so this is the residue, isn't it? The residue is substitute here. See here, 1600 is the total. So 256 into 100. You will get it. Which one, you know? 60. Just strike out 16 into 16, you will get 256. Okay, 16%. Okay, 16%. 16 we will get it. This one is what is that, you know? Q squared is the video already you saw it. Suppose if you want, just go to the just type the question paper in the Google, you will get the answer. The video will display, and just go through it, you will get the ideas and also answer it. So here, which one, you know, Q squared, that is equal to 16 means Q is equal to, what is that? So this is the root. So you will get 0.4. Okay. So 4. This one is Q. Likewise, you have to calculate which one, you know, P also. So the 0 is 4% is that. What about a Q? Q is also, you have to add another point. So 256 plus 256. How much is that? See here. 256 is the Q square, isn't it? Q square, isn't it? So P squared is also same, isn't it? P squared is also same, like this. Okay. So 256. So add these two, P squared plus Q squared. What will happen? So you will get 700 and something, isn't it? 256 and 256. Add 2, 1, 1 is the 512, isn't it? 512. 512. Okay, you have to, this one is a total of which one, you know, P squared as well as a Q squared. We need which one, you know, PQ, isn't it? PQ calculate means what can you know, you have to minus it, this one, from the this total 2PQ. So exactly, I don't know exactly the calculation is seen here. So I will show you next time, my dear students. I will show you, I will work out it and then I will tell you. Sorry for the inconvenience. Okay. So the max is okay. I think that's all about this part, I think. So next part is also there. So we'll see the after the calculation. Okay, I will next class I will show it that calculation. Okay, the video you have taken, but it is not playing. So okay, but the question part is just we discussed. What is by Turner syndrome in is due to the which one, you know? It's nothing but the monosomic chromosomes. Already we discussed. Turner syndrome is also due to the monosomic chromosomes. Okay. So this one already we discussed. Which one? 45 plus XO, isn't it? Turner syndromes. Allosomic is not possible. Trisomic is not possible. Tetrasomic is or not possible in this category, Turner syndrome. Okay. What about the next one? See that the best examples of polyploidy. Okay. So the ABO blood grouping is not Varad. It's not coming because it comes under the headings known as what is that? Multiple alleles, skin colors. It's all a phenylketonuria. So phenylketonuria is an example. Best examples are pleiotropy. Sorry, my dear students, pleiotropy. I'm not a pleiotropy. What is that? Phenylketonuria is a pleiotropy examples. Okay, so go to the next one. I will explain it in deeply. See that in cross between a male and a female, the both heterozygous for sickle cell anemia. See here, the cross between a male and a female, both heterozygous for sickle cell anemia gene, what percentage of the progeny will be diseased? See, both are heterozygous in nature. That means a F1 generation. See here, this is a HPA, HPS is also there. This one is also HPA, HPS. So both are heterozygous nature. The parent is heterozygous, and also what happened, heterozygous for sickle cell anemia because the S is present. It is. So sickle cell. So what will be the progeny? How many percentage is possible? Like that they ask the question. You have to divide these two. Also here, tools also. The correspondingly, it may be, what is a cross means you will get it there just like a dihybrid cross, 1:2:1 ratio. So the progeny will be how many percentage mean 25% is possible. Okay. Can you see this one? F1 gen into F1 like that. Just imagine F1 into F1. So you will get it two gametes. Okay. So fuse together. Okay. You will get it 1:2:1 ratio. The one is similar to the parent, two is a what is a affected one. Okay. And then last one is also what is a recessive character is there, similar to that of the parent. But here what they asked, gene, what percentage of the progeny will be diseased only they asked. So only the 25% of the disease will be possible. You have to put the calculation, then only see that, see here, see there, the solution. So this is the parental characteristics. The correct option is C, 26%. Explanation of the correct option is according to the questions, step 1, the formula used. What will be the formula means the percentage of the affected individuals that is equal to number of affected progeny divided by total number of the progeny into 100. See, this is the first step. You have to use the first formula. This is the formula. Percentage affected individual. Number of affected progeny. How many numbers? Okay, suppose 2. Okay, total number of progeny 4 into 100. See, the number of affected progeny is 1. Okay, number of affected progeny is 1. The total number of progeny is 4. See here. So these are the 4 progenies we will get it. From this one, the total HPS is the affected one. So these are all are carrier, isn't it? This is a recessive character. These are all are heterozygous character. This is the affected one. So the 4 progeny is there. You will get it only one only. So this is the 1 is a affected one. 4 is a progeny. Applying the formula, what will happen? 1 divided 4 into 100, you will get it 25%. That is the answer. Any doubt in this area, my dear? Ajay?
No, sir. Yes, sir.
Oh, do you have any idea, doubt in this area?
No, sir. No doubt, sir.
Once again. Are you clear now? This part.
Yes, sir.
Yes. Okay. See here, once again, these two are heterozygous parent, isn't it? Heterozygous parent, isn't it? See, the heterozygous parents, this is the F1 generation, and you will get the F2 generation. There are 4 progenies possible. Okay, so you have to put the checkerboard. Okay, suppose we are seeing this type of question, just to put the checkerboard. Okay, then only we will get the progeny result clearly. The first one is HPS, both are homozygous, it is affected. This is similar to that of parent. These two are similar to that of the parent. This one is the non-affected or unaffected. Okay, so you have to use the formula. Which one is the formula? Percentage of affected individual, that is equal to number of affected progeny divided by total number of the progeny. Total number of progeny, what will happen? 4 is there. Which one is affected? Only one is affected here. So this is the one. The progeny is affected progeny is 1. The total number of progeny is 4. Applying the formula, you will get it 1 by 4 into 100. That is equal to 25%. The answer will be the fourth one. Okay, so this is the things. And next one is select the correct matches. Already we studied the hemophilia, okay, and phenylketonuria, sickle cell anemia, triads, and thalassemia. These are all are what is the name? Some mostly they are all are X-linked. Okay, so you have the doubt in this area. Which one is you can easily fix it? Will you please? Which is correctly matched? See the solution. Okay, see the solution. Phenylketonuria is an autosomal recessive triad. Note it down, my dear. Phenylketonuria is an autosomal recessive triad. Thalassemia is an autosomal recessive disease, gene present on chromosome number 11. Hemophilia is an X-linked recessive. This is autosomal recessive. This is the 11th chromosome. This is also a 11th. See here, the hemophilia is also X-linked recessive disease. Sickle cell anemia is also autosomal recessive triads present on the chromosome number 11. So thalassemia as well as the sickle cell anemia is also, both are same. See the autosomal recessive here also. This is also autosomal recessive. Okay. See the 11th chromosome, both are same. Okay. So the correct answer is sickle cell anemia, autosomal recessive triads, chromosome 11 is the correct one they have given in this part. Okay. And go to the next one is called as, what is that? What map unit? Centimorgan. Centimorgan is nothing but the unit. Construction of map, gene map is also measured by the, which one? Centimorgan. Is adapted in the construction of genetic map. Which one is the correct one? How many percentage they asked? How many percentage means the unit of distance between gene or chromosome representing 50% of crossing over. It is not possible. How? The unit of distance between the two expressed gene representing 10% crossing over. No. The unit of distance between the two expressed gene representing 100% of crossing over. So these are all are same. The percentage will be differ. Okay, 50%, 10%, 100%. At the same time, last one, the unit of distance between the two expressed genes representing 1% of crossing over is correctly matched. This is the correct one. Okay, how will you construct the genetic map? During the calculation, during the genetic map constructions, it will not possible. How? When the distance between the genes of chromosome the increases, what happened? The crossing over increases. Suppose if it is near, what happened? The crossing over is completely loosed. Already we studied, isn't it? So that is the things. The largest gene chromosomes known as human gene is known as dystrophin. The largest known human gene is which one? Dystrophin. Note it down. This is not atropine, tropin, and ditropins. You have to note it down. This one is the correct one. Dystrophin. Okay. So the largest human genome. See that hints has given. So note it down. This is very big explanation has given here. So note it this one. Just note it down. So additional information. See here the dystrophin associated protein are often divided into 3 groups supporting to their cellular, localizing, extracellular, transmembrane, cytoplasm. These are all known in. Just note it down. Which one is the largest high gene human being? Dystrophin. Okay. So I think this part is to finish it, and remaining things is also similar to this one, and the part 7th one. Okay. So just I gave a small intro only, then we will enter into the next class. See this one. So this is another one. Okay. So these are the another type of which one? The principles of inheritance follow continuity. So this one, what happened, so another, what are the metabolic disorders also takes place in it. So already we studied that things also. This is the glucose 6-phosphate dehydrogenase deficiency. Where will you study this one, my dear students? Glucose 6-phosphate. Do you know any idea about this one? Ajay? Do you know? What about glycolysis?
Sir.
Yeah.
Sir.
Glycolysis.
Glycolysis, sir.
Yes, glucose converted into glucose 6-phosphate.
Sir, glucose 6-phosphate, sir.
Yeah, that one. Okay. See here, I will show you simply, then we'll finish it. Okay. See this one, just for intro only. Because, so already know about glycolysis. Okay, so glycolysis, about glucose molecules split to gives, what is that, glucose split to gives glucose 6-phosphate. With the help of the enzyme is called as, what is that, hexokinase. Hexokinase, isn't it? Kinase. Then glucose 6-phosphate converted into fructose 6-phosphate. Fructose 6-phosphate converted to fructose 1,6-bisphosphate. And then fructose 1,6-phosphate converted to dihydroxy acetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, like that it converts. Here the isomerase enzyme is also there. The glyceraldehyde 3-phosphate converted into pyruvate. Here also the 5-step is there. Here also 5-step is there. 10 enzyme, 10 byproduct is also formation takes place. First ATP utilization is also there. Okay. So this is the things as given here. So we'll see these things in the next classes, how it will be like that. Okay. I will finish it up to this level. Okay. Okay, my dear students, we will finish it up to this level. So we'll see the remaining things on the next class. I will show you the next two classes, the calculation aspects. Okay. Okay. Thank you. Thank you, my dear students.
Thank you, sir.
Thank you, Pa. Thank you.
Thank you, sir.
Thank you, Ganna. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you so much.